So, okay, my pace at the moment is I train somewhere between 5 and 6 minutes per hour. I should run slower than I do, but I'm very competitive and too much into numbers. So, but at the moment, I let Garmin coach me, and my half marathon goal time is 1 hour 54. But yeah, let's see where it brings me. And yes, I wish I rode less and studied more, because I did attend the classes, but I was mostly thinking about rowing. So yeah, I'm quite competitive. I also played chess with my partner, and recently we also had a fight for 3 hours about chess being not a game, uh, and because I won. So I guess I lost the next time, so it kind of restabilized our relationship. So you see kind of maybe where I'm going. Like, I'm very broadly interested. Originally, I wanted to work in the International Court of Justice in The Hague, but now I'm here. So I think, I really like having a group, working with people and helping them find their way. And I always struggle a little bit with like finding the connections of my questions, which I think there are, but like with Klaus, we also spoke about like how to filter out the true residual of what you're trying to say, also in terms of like wordings in neurosciences as cortical hierarchy and inside outside. So yeah, this is one of my key struggles, to start it like as a safe space. And today I will talk to you a bit about the cortical tapestry and how I see the brain kind of as the nexus between like the individual and the society we live in. And to me, this is also one way, to understand how the brain functions as it does. And I'll just go back in with my slide in. So yeah, the model that I really am kind of like interested in, and this is also at the moment also quite inspired by my partner, who started as a child and adolescent psychiatrist. So this also re-humbled myself about all the brain science that we do, because in the end, if you think about people with mental health conditions, it's not only the biology that matters, but also the circumstances that you're in and the chances that we're all getting, right? So this is also one of the translational things I have now more prominent on my mind. So yeah, I find this question, like what is this interrelationship between the individual and their social surroundings, super fascinating. At the same time, like you guys, I like coding and networks and understanding rules in the system and like latent dimensions. So this is more my day-to-day activity. And like just now, I was also coding to figure out whether I made a mistake, or maybe my student can relook at the code again. So I think you know how it is. So yeah, the brain. Well, this is not the whole brain, it's only the cortex, and I will get back to that also at some point. It has a certain function in it, and this is enabled roughly by genes, right? This genetic program builds a brain, gets a brain that has a certain features. However, this cannot work without an environment. We need input to function, like whether it's from the body, but also from our surroundings. Thinking also about the visual system is built up through activity-guided plasticity. So it's a circle, and even more so, it matters like how the brain is wired, what genes and how the genetic expression is relevant for the brain to develop, and like in what context certain features come out, also in the context of mental health, and also thinking about like how we think about behavior and its individual variation per se, right? So it's like this eternal circle where like both things interrelate with each other in a very broad sense. So it's a bit of a mess, right? And our task is to kind of like create order in this chaos, at least at some level, by combining different skills and approaches. At least that is what I do. And here, my hammer and nail is computational approaches and my computer. And this is even more so the case for my students, who are all much more proficient in like any type of coding than I am. So, well, I don't see my own title of the slide, but I'm probably saying something about brain structure as a coordinate system. So like one of the questions or challenges, if you have like big and broad questions like I do, is where is my anchor, right? Like where is my lighting pole? And to me, the anchor is actually in the organization of the brain, because this can provide a coordinate system to make maps and to help us find a way. And here I start often with anatomy, an anatomical hierarchy. And I should now have edited this slide and saying more like this inside-outside view, because it's only like a sequential line and not a hierarchy of systems and systems, although this is also present in the brain. And here you can also measure this in vivo using, for example, T1, T2 weighted ratio, although this is not the most optimal mapping feature of myelin, like MT or quantitative T1 are actually much better. Then we also can measure this using function by doing, for example, dimension reductions techniques and thinking about latent dimension in our function organization. And then last but not least, this also may relate to a differentiation of how regions have expanded as a consequence of their evolutionary role, thinking also from a multi-species perspective, like we have humans, right? And if you want to understand variability, we can also look what else is out there on the planet. And this also may help us understand certain rules of how the brain and the genes are shaping or genes are shaping the brain and how they support the functionality that we have as humans and the specializations that we have as humans relative to other species. So when I started my postdoc with Simon Eickhoff, he was like, yeah, structural covariance, you've worked a bit on it. Yeah, maybe think a bit more about it. Like what does it mean? Like there's this association, but it's a bit of genes, a bit of environment. So maybe go figure something out with it. And yeah, I love structural covariance because, yeah, also with Klaus, we spoke briefly about it. It's in one way a kind of a memory of the past, right? Because these regions, they covary because they're similar genetic, ontogenetic roots. So when the brain is built, the structure of these regions is shaped and is co-shaped, and that's why it varies across a population with each other. So this is like an intra-individual measure. So you correlate, for example, the dorsal anterior insula of us all with the supramarginal gyrus of us all, and we see a positive correlation. So when one gets thicker, the other also gets thicker or thinner. And you don't have this in all. So you create kind of networks. And then I used a twin model comparing monozygotic and dizygotic twins and unrelated individuals to see if this covariance is stronger as a function of genetic similarity. And the second model that I used was a comparative model comparing humans to macaque monkeys to see if this covariance profile was actually similar in humans and these other primates. And then I also used the gradient approach, which is a low-dimensional reduction technique. So first you get an input matrix, then you create an affinity matrix using a kernel function, in this case, a cosine similarity function. And we did nonlinear decomposition, thinking about the first 10 components. Mostly we look at the first 2 components because they explain most variance in this data structure. And then you have an ordered axis based on the patterns of the matrix. So in this case, it would be an ordered pattern of how similar regions have covariant profiles with each other. So then, like, to understand this organization of covariance, I did this diffusion map embedding, although principal component analysis would have given, which is a linear approach, would have given the same or a highly similar pattern. And I saw mainly a differentiation between posterior to anterior regions with a similar color showing a covariant pattern in humans, but also in macaques. Although macaques, it's slightly different, and this is in part because of the parcellation, in part because the N is much lower. But the upside is I'm super grateful that this open data is shared through the PRIME-DE dataset, because this made me able to check this out in the first place. And actually, this relates to the differentiation or this inside-out pattern or gradient that Klaus also likes to talk about between sensory motor and more transmodal or inside regions. At the same time, we also saw a more superior-inferior pattern here in red and green. And this was also more or less present in macaques. And we also did spatial correlations by mapping the human into macaque space with an algorithm that Ting Xu had developed, where she had like these anchors. And with this, you can kind of morph the connectivity space of one species, or you can only go down mostly, because the other way around is tricky because certain regions are more expanded. And they also show like statistical similarities in their spatial variation. And this actually related, and also Alexandros Goulas, who was also here, wrote a very nice paper on this, with the dual origin model, suggesting that there's different waves of laminarization coming from the piriform cortex, which is also part of the olfactory system, and the archicortex, which is mainly the hippocampus. So in a way, one could say that there could be there are more, but two main axes of genetic organization in the cortex. And in this way, it kind of provides a compass or a map of the different dimensions one can think about when thinking about how structure is covariant and organized. And this differentiation can be kind of made up of different anchors, so to say, mainly also the dual origin, but also this inside-out view. Then the question also becomes like, how can this structure then support function? And like we spoke also about Helen Barbas this morning, so it's all returning a little bit. So on the one hand, there's a structural model that says that microstructure relates to connectivity, and here it means that like connects to like, and that if you go up this differentiation or down this differentiation, one could rather say, one also sees feed forward and feedback connectivity profiles in terms of how regions are connected. Another idea, which is not quite conflicting, but also not quite the same as well, is the idea of the tethering hypothesis, namely that you have like signaling cascades going from primary areas, and based on this, you create also a network where you have ultimately a self-organizing network at the peak of distance of these signaling cascades, so that kind of the function gets untethered from these more signaling cascades that come from the outside, and that this is how then ultimately function is shaped upon these structural constraints. Then I also wondered, like, how does this then relate to functional maps and functional processes? Thinking about this dual axis with, on the one hand, this inside-outside view relating from coupling to uncoupling. And indeed, you see that outside regions relate, or coupled regions, relate more to motor, auditory, attention. But at the same time, if you think about the second axis, namely the differentiation between piriform and hippocampal areas, you see that this differentiate more social cognition, episodic memory, attention. So maybe one could say mnemonic cognitive operations from emotion, pain. Semantics is also there. I don't know, but what that means, but like the rough interpretation could be that one also has like different forms of thinking that are associated in this two-dimensional space, also differentiating more cognitive versus emotion, motivation ways of processing information. And there's actually, I then got a small obsession also with the olfactory cortex and how underappreciated this is in humans. And there's also a philosopher working on this, like smellosophy. The book is called, and this is about like how do we think about thinking and how this is shaped by maybe visual processing, but like that other ways of perceiving our world, such as olfaction, may give us also different hints at alternative codes of cognition, if you may say, so that there's not one way of thinking, and we should actually look at all our senses to really get at that. There's maybe different ways that our brain works in relationship to how we think, and not everything is this one route. So together, it could be that it creates a more or less cognitive landscape embedded in brain organization and ultimately supporting flexible cognition through having these different anchor points. So then my lab started, or it had already started, but I was still wrapping up my postdoc projects because I think it's always good practice to also be first author, even if you're last author. And I also had to do this still because I promised. And with Amin, we started to think about, how this covariance idea relates then to microstructure, namely the thickness of laminar sheets. So as you see here, and as you probably all well know, is that roughly, or most people, if you asked and they know something about neuroanatomy, they would say, the cortex has roughly 6 layers, especially the isocortex, the agranular archicortex, that's a little bit more tricky, and also parts of the motor cortex also miss a little bit, or it's less clear, but overall, most part of the cortex has 6 layers, although some other people then subdivide these layers even further, but this is a model that we didn't incorporate. These layers are horizontally superimposed, and this is something that has been long known. And it has been said, or Van Economo already, and also Garcia Cabezas, said that there's different types of cortex depending on their structure. However, these models and like these domains of cortical types are largely theory-driven and they have a low sampling rate. However, what if we use the whole brain or the whole isocortex and do a data-driven approach of these different cortical types based on their laminar profiles? So we had this big brain of Katrin Amons, which is a 65-year-old male. He passed away, otherwise we wouldn't be able to slice his brain at a super high resolution. Cell bodies were stained, and they created an ultra-high resolution 3D atlas at 20 micrometers. Then there was a layer segmentation using convolutional neural networks, as you see here in the GIF, like it slowly changes from gray to the lines. Oh, and volumes of this segmentation were projected on the surface. Then Amin, who is amazing, he did pre-processing, so we had these layers. And we first excluded the A and this granular layers because there the layer segmentation obviously wouldn't make so much sense because the layers are less clear. Second, we did some smoothing to get rid of the Bock effect. So I don't know if you've heard of it, but it means that the thickness of layers is different for the sulci and gyri. And henceforth, we didn't want to create a map of like gyrification. So we smoothed it, as you see here. The difference between sulci and gyri was massively reduced, which is what we wanted. Then we normalized it by total thickness. We applied a parcellation to make it more computational scalable. And then we had a relative laminar thickness in each region, as you see here. Following, we created a partial correlation to see if the relative laminar thickness of a given region covaried with the relative laminar thickness of another region. And we performed a PCA to see what regions would then have a similar laminar thickness covariance profile. And as you see, it was mainly a differentiation between posterior regions, excluding V1, towards more lateral frontal regions, here in green-blue, as you see here. So what does this pattern then mean? So we then correlated the pattern with the laminar thickness profile, and we see that it's a differentiation of more superior and inferior layer thickness, going from a thick layer, what the blue regions, sorry that I say it right, so the blue regions, so a negative correlation, or they show highest thickness in the deep, like layer 6 in the, the blue regions show the thickest layer 6 and the yellow region, so the thinnest layer 6, and this pattern was reversed for layer 3. So it shows a differentiation between supragranular and superior and inferior thickness patterns. Then we also wanted to see like how would this then relate to social covariance, which is like, as we know, this group level map where it's like more of a global cortical thickness pattern. And indeed, overall, if we take all the nodes, we found a correlation of 0.33 and a significant correlation between this laminar thickness profile, which is just based on one individual who was also old, versus the social covariance we see in a general population of the Human Connectome Project. And indeed, if you then also have the principal gradient, and this is the exact same gradient as I showed you earlier of my study, and this laminar thickness gradient, there's also a spatial correlation between both. Although the correlation is not perfect, it's 0.55, so at least it explains in part covariance. Then we wondered also about average laminar density and cell size. So we combined the principal axis of laminar thickness and density. And as you see here, this is highly similar to the laminar thickness pattern. And as you see, if you look at the principal axis bins, yeah, you see my mouse, it is like the blue regions have a least dense profile, and it gets darker and darker. So more and more cells are in the more posterior regions, as you would have also expected. And this is particularly true for layer 4 and 5, but actually for all layers, there's a negative correlation. And about laminar thickness, we already discussed this. There's a differentiation between primarily layer 6 versus layer 3. Then we also wanted to think about cell size. And for this, we used a sampling method from Erik Opschulte, who made this data openly available, because the big brain is like a lot of people work on different questions on this brain. So there's also a lot of data available to like compare to. And you see here also with the convolutional network and different steps, these cells are also segmented and like measured for their size. And you see that there's like different sample locations. And one of the downsides is that they're not that many, but the upside is that they are there überhaupt, and also that we then can relate this to our gradient. And what you see is that this pattern of cell density and cell size corresponds negatively, particularly to layer 4, but also to layer 3, and for cell size, it also corresponds to layer 5 and 6. And indeed, also the pattern that we see of the laminar thickness covariance relates to external pyramidization, which is the differentiation between neuronal size in layer 3 and layer 5. So as you see here, the correlation is not that strong, and this could be in part also because of the sampling, and more work needs to be done, but this aligns pretty nice with also other studies in non-human primates. Then, like in line or in the vein of Helen Barba's model, we wanted to see if there's a relationship between laminar structure and also feed-forward and feedback projections using indirect measures. Because on the one hand, we had an asymmetry-based functional hierarchy, as we called it, and this is based between the difference between weighted out and weighted in degree. And here, indeed, we saw a correlation between the laminar thickness covariance gradient in this asymmetry-based functional hierarchy. And second, we then also, because in macaques we have feed-forward feedback profiles based on track tracing, but we didn't have this laminar thickness profiles. So we projected the data from human to macaque and then compared it using this different model of feed-forward feedback profiles. And indeed, also here we saw this spatial relationship. So this is, and also one of the reviewers commented, it's kind of interesting, but it's also quite of indirect evidence, right? So this calls for more studies that maybe look at this laminar architecture and then try to combine this with functional data. And this is also something we're doing now with somebody else, where we have actually in vivo Zeventesta data, and we may get a more one-to-one mapping of these laminar profiles and functional maps using resting state. So interim summary would be that there's a link between group level macroscale cortical thickness covariance and individual layer thickness covariance. That layer thickness covariance organization differentiates layer and cellular features, and that this has a spatial association with functional processing hierarchies inside eye profiles axes. And yeah, there is a note also, and I think if you read the paper, you see also the discussion is very long. Because one question is also like, what these spatial relationships, what do they really mean? Is it maybe mostly about distance between regions and not so much about cortical types? And this is something that it seems that cortical type is very relevant in organizing this whole thing, but distance is also very closely related to this matter. And this is actually an ongoing debate also to understand what shapes these connections. And yeah, we try to accommodate the reviewer in the discussion, and we learned a lot about like all the literature that is already out there and also has been done in non-human primates. And yeah, it was a it was very cool. So then moving forward together with Maaike, we wondered, like, could studying the organizational principles also provide valuable information on the co-occurrence of structural alterations? So as you know, there are overlapping risk factors in neuropsychiatric and neurodevelopmental conditions if you look at the genetic correlations between these traits. Moreover, there's overlapping endophenotypes if you look at the meta-analytical and functional, but also structural maps. Then we know that many of these pathologies are network-based. So it's not that only one region is affected, but actually it relates to the connections a region has with the rest of the brain. And last, and this is also something that helped us very much in the pandemic and in general, like consortia and open science make it possible to connect different data sets with each other and ask questions to build bridges and like integrate them more. And this is what I like. So what we did is we had like 6 disorders / conditions and had the Cohen's D cortical thickness map. So we had bipolar disorder, ADHD, autism spectrum condition, depression, obsessive compulsive disorder, and schizophrenia. And all these maps indicate whether there's relative decreases or increases in cortical thickness relative to their control cohort. And this is all based on Enigma data and openly available in the Enigma toolbox. So first we wanted to see what happens if we covary this and see if impact of disorder is shared across disorders. So say that a certain, like prefrontal cortex has a high impact in schizophrenia and less so in autism, is this then the same for parietal regions, so that there is like this disorder-based covariance? So we made this correlation matrix, and then we studied co-alteration hubs of regions that show a lot of covariance with each other. And you see this is mainly, or it's almost all the brain, but it highlights also frontal cortices, but also temporal regions, and not so much posterior regions. And then we used an approach which is called epicenter mapping. And here you see how this hub map relates to the connectivity profiles of specific regions, sort of trying to figure out, like, is there like a connectivity profile that can explain these local changes? And there is, and in particular the connectivity profiles of function, but also structure in association areas, so temporal and parietal and frontal areas relate to this hub map, as you see here. So as we also knew already, but here we showed empirically, like what is shared across all these disorders is like maybe this connectivity alteration, and particularly in these regions that are highlighted here. Then what I was also curious about is like, what are then the organizational principles that underlie these conditions? And for this, we used the gradient approach to see what spatial variation explained most variance in this cross-disorder co-alteration patterning. And indeed, here we see that there's a differentiation between posterior and temporal areas and frontal areas on the one hand, and the second gradient explained a differentiation between posterior regions, so parietal and also occipital regions, and superior temporal areas mostly, but also some motor cortex. What is nice is, again, like similar to this laminar thickness covariance, it relates also to group level covariance of healthy individuals. So the impact of disorder that is shared across disorders also relates to this social covariance per se, in particular gradient 1. And it also relates to shared disruptions in different levels of functional processing, differentiating sensory, attention, perception, and cognitive control, roughly. So it suggests a potential independent maturation or cause and distinct vulnerability windows for different functional processing levels. So then I extended this a little bit because I was curious about addiction, because substance dependence is characterized by a loss of control over various drug and alcohol intake and behavior, which contributes also to high relapse rates. However, the biomarkers are quite unknown, and it's also hard, right? Because using these drugs also impacts the brain structure, and henceforth you get this cascade. And then ideally, we want to provide a treatment on the basis of an individual's patient vulnerabilities. But then the question comes, like similar to the previous work, how does then addiction impact the brain in a systematic way? So here I combine data of alcohol, cocaine, amphetamines, nicotine, cannabis, and opioids together. And again, you see that these hubs, where you see a lot of covariance across addictive subtypes or substance use disorders, are in frontal areas, but also sensory areas, as you see here in yellow. And the pattern is rather nice, symmetrical, which is often a good sign. And what I found kind of interesting is that if you correlate this with normal social covariance, you see that overall these disorders or substance use seems to follow the organization that you would expect, namely that of covariance, but not so much in orbitofrontal and lateral frontal areas, maybe also suggesting that there the pattern is kind of broken, maybe because of the use of these drugs and alcohol. But yeah, I'm still working on the project, so I haven't really made up my mind, but I found it interesting that the rule apparently doesn't apply in these orbitofrontal areas that are also often impacted with substance use disorder. And indeed, we see the same pattern if we differentiate it also from neuropsychiatric co-alteration, but also general structure covariant. Then if we made a gradient, and I think this also brings home the point, is that you see this strong overlap spatially between normative covariance, which also relates to development, and neuropsychiatric co-alteration patterns. But for substance use, it is different, and it kind of differentiates this orbitofrontal from the rest of the brain in the first gradient. And for the second gradient, you see, again, like a bit of a messy pattern, in fact, but it also differentiates orbitofrontal, but also parietal areas from frontal cortices. And for neuropsychiatric, we see actually a different pattern happening. And if you combine it there, this is me trying to understand what is going on, and I'm not quite through it yet, but what I kind of found interesting that it seems to be, but this is maybe more in the eye of the beholder than the statistics will tell, but that the differentiation in this neurodevelopmental condition is mainly about this first gradient that relates to development and this differentiation between posterior and anterior areas. But actually, if you think about what is happening in substance use disorders, you see a differentiation between this inferior or maybe MOFC, like addiction, orbitofrontal areas versus superior areas. So this is now something I'm trying to understand, is how different axes may also point to different neurobiological associations with these conditions. Then in the second part, we're also nearing the end, but I wanted to talk about plasticity and adaptation because we shouldn't think about conditions and in general about the brain as something stable, right? Because it changes all the time and we have different brains, but we also will have different brains 1 year from now, 1 day from now, 1 week from now. So it's a very it's an ongoing process that happens on different time scales and is part of development and adaptation and also of aging. However, how the brain anchors this change and what stays the same and what changes is incompletely understood. But it may again be that this system-level organization and structural variation of the brain may show specific changes and may kind of show a principle of how the brain can change and where it changes. And this may then help to build interventions and understand vulnerable time windows in the brain according to its development and the type of system subset that is impacted. So with Amin, we're now working on adolescent maturation using individualized biophysical network modeling because, well, as we know or have experienced, adolescence is a critical period of development with substantial macro and microscale changes in the brain, including also the maturation of cortical microcircuit and changes in excitation and inhibition balance. However, studying this in vivo is a challenge in humans, but biophysical network modeling is a promising approach. So here we use an individualized model where we try to see how EI changes in adolescence over time in 2 different data sets. So in short, we fitted the model, which is a balloon and a watershed model. No, what is it called? Okay, now I forgot the name. It doesn't matter so much, but we fitted it on the functional dynamic model on the one hand, and also the functional connectivity. So we had, like, in terms of functions, 2 operationalization, one dynamic and one stable functional connectome. And then we measured the goodness of fit. And we did, using clays, we did model optimization. And then we extracted excitatory firing rate and the optimal simulations. So what we found in the PNC data set is actually that there is an association between age-related change and changes in the excitation inhibition balance over the course of between 12 and 20 years old. And it's a nice differentiation again between sensory and more prefrontal and parietal regions. And also in the imaging data set, which is a longitudinal design, we found a similar pattern, which is really nice because these models are very highly parameterized and very tricky to set up. And one often has the feeling yeah, what is then the ground truth? But we're very happy and we're doing way more checks than this, but that we find at least a consistent pattern in 2 different data sets and 2 different approaches related to aging. And we find that it decreases in association areas and increases in unimodal regions. And this also relates to previous animal and human studies showing a decreased EI ratio in association areas. And it is possible that this individualized modeling holds promise for future interrogation of the interrelationship between this microcircuit maturation and cognitive as well as clinical variation. So this is ongoing work. And for this also Amin, he developed a GPU code to optimize this individualized procedure because the CPU was too slow. And we're now trying to understand also how this relates to functional changes and dynamics. And we hope to have the preprint out soon. Another element where we also were wondering about and what we try to understand is on the one hand, we show that there's this relationship between brains that have of people that have a condition or disorder diagnosed. But what if the brain is also something that makes a human more resilient? So how do we understand resilient change over time, and how does healthy development then look at? So here we looked at, we created a measure of resilience, which means like how much better are you feeling relative to the adversity that you've experienced. So you can only be resilient against something that has happened to you. And this is a marker that we then created also in an adolescent sample. And this is an out-of-sample marker where we then uses the weight to create this resilient factor for imaging sub-study. So we related this to myelination, and what you see is that individuals that have an increased resilience over time show also an accelerated maturation in prefrontal cortex of a myelin proxy. And this also coincided with an increased connectivity of these regions with especially association areas, namely default mode and frontal parietal networks, meaning that maybe in those individuals that are more resilient, they also age a little bit, like their brain matures a bit faster relative to those who don't. So it seems that developing towards a more resilient psychosocial functioning was linked to a higher rate of estimated myelination in the anterolateral prefrontal cortex. And it seems that the functional connectivity seems to be more maintained. However, what is also interesting in terms of like system level changes is that we see a stronger reorganization in people that are more resilient. So it seems that like these more psychological concept of feeling better despite your adverse experiences also relates to how these myelin networks mature and reorganize over time. So it's a very dynamic relationship between individual differences and maybe also a buffer, like protecting you for stressors or mental health. Last, I also want to talk a little bit about intervention. And like maybe start off with, like, I speak about this cognitive elements, and maybe a lot of it is more indirect and links meta-analytical maps to each other. But one can also think about this model, and this is more from, so we're switching gears a little bit, apologies for this, but I don't know if you meditate or like are aware of like to me, sports is also a form of meditation, but you can also think about the states that you can be in your mind as a sort of a room of a different like roles that it has. So on the one hand, you have like a more meta-awareness, but you also have object orientation. And in this room, you can have different states and spaces, and this you can just probe with mental training by guiding people to be in a certain state or position in this room. And this may relate also to the brain organization and adaptation. So together with Tanya Singer, and this started in my PhD, but I'm now finally wrapping up the last of my PhD projects, we trained different social emotional skills through mental training. So on the one hand, we focused on attention and interoceptive awareness. A second training was more about compassion, prosocial motivation, dealing with difficult emotions, so more this affective domain. And the last part was more about perspective taking, so having more cognitive ideas about the different roles that we have in our life. And this was done for 3 times 3 months in a design that I will show you here. So we had 2 cohorts, and they underwent 3 types of mental training. And the first hand, it was like first learning how to do mental training in the first place. And this was more in this attention mindfulness block in yellow. And then a crossover design, differentiating more the social affective versus social cognitive skills. And then we also had retest controls. So my idea was like, what if all these functions also relate to different architectural architectures? So for this I took the meta-analytical maps of attention and interoception versus emotion, empathy, and theory of mind. And as you see here, if you look at intracortical microstructure as measured with a quantitative T1, each of them showed a slightly different, although overall, of course, a differentiation between superficial and deep layers. Each of these functions also has its unique profile, and maybe this also relates to how these functions work and how they relate to each other. And it also showed a slightly different overall embedding of functional organization. And indeed, if you compare presence, so this attention, mindfulness versus perspective taking, you see that overall networks become more differentiated when you do this present attention mindfulness, but more integrated when you do this perspective taking tasks, but they remain more stable when you do more this emotional motivation. And this may also relate to the different functions it is associated with. However, what I find interesting, and this is also something that I want to work further on or try to understand, if you look at the changes in microstructure, also as a function of that, but also in general, you see a differentiable pattern if you compare the more attention versus the cognitive versus the emotional motivation axes, where you also see that deeper layers seem to also be impacted mainly in this social affective module, whereas the other layers show more differences in superior, or the other training programs show more superficial changes. So it seems not only that the space matters, but also the depth of the process of where you expect it. And a big side note is that this is, of course, only done in 3 tester data, and it is to some extent, like the power is pretty high for this kind of study, but it does show that one has to think about plasticity not only in where it's happening, but also how it's happening in the brain and how a certain region is changing, and thinking beyond changes in thickness or BBM, for example. And what I also want to say is also in relation to, like, a lot of my studies point to changes in these default mode frontoparietal areas, and actually the most robust change that I observed in both training cohorts is this overall change in these blue regions. And here blue means increased myelination. So in particular, over the 9 months time, we see in middle but also deeper layers this increase in myelination, yeah, as you see also here depicted. So 2 notes before I'm done. Sorry if it was a bit much, but I think one thing that is important to keep in mind is that there's also sex differences and neuroendocrine factors that play a significant role when thinking about the brain. And in particular, these hormonal transition periods are also important factors thinking about mental health challenges, both in men and women. So together with Svenja, we looked at microstructure, and thinking also about the structural model of plasticity and stability, we wondered if those regions that are agranular and are supposed to be more plastic are also those regions where we see changes as a function for hormonal status, and where we took an indirect approach, namely the days after menses start, and then we kind of reconfigured what menstrual stage they should be in and what hormones should be high and low, and also compared this to oral contraceptive cohort. And what we see is that there's marked changes in overall mean of microstructure as a function of menstrual state, but also if you compare males versus females that take oral contraceptives or also are in low estrogen phases or high progesterone females, you see different patterns and different differences between males and females as a function of the hormonal stage. So maybe these microstructural markers can also tell us a little bit about, like, different variations related to neuroendocrine markers. We did check also to potential effects of vasculature, because it's also water that changes, right? But their findings did seem to be robust. And also when doing a spatial correlations of where we observed most effect and gene expression patterns of these sex hormones, we found again a similar pattern, suggesting that, like, maybe something is really, like, adapting also as this circle of plasticity, or this month of menses. So this menstrual month is continuing. We see also, like, these tides changing in cortical microstructure. And then Bianca and Denise is actually a high school student from Palo Alto, and she wrote her first paper with us. And well, this is mainly Bianca's job, actually. Like, I'm so proud of Bianca. And they looked actually at an open data set where they looked at the functional connections, and they have every day a marker of hormonal changes in men and in women. And then they correlated the changes in hormones with this day-by-day fluctuation in functional organization. And indeed, they found also a relationship with testosterone in the male axis of the inside and the outside of the brain, so to say. And they also found global effects of estradiol, progesterone, and perceived stress, mainly also in these limbic areas and frontoparietal control networks. And similar but different for men, they also found association between testosterone and cortisol effects, like arguing that both men and women have different but yet also show this co-fluctuation as a function of, on the one hand, stress and cortisol, but also other hormones. Oh yeah, and then a very, very last note. Sorry, like it keeps being like more, but yeah, the cerebellum has also been a region, a part of the brain that is very awesome. And the people that study it are also amazing, because the interesting thing is that, like, for the cortex, we know we have cortical types, and there's all these kind of theories on it. But for the cerebellum, we don't really have cerebellar types, because there the microstructure is rather homogeneous. Although now people are arguing that it's not that homogeneous as has been thought. And we have these different lobules. And what is interesting also is that there's this functional differentiation in the cerebellum, and that it's not only a motor-related region, but that it also relates to social cognitive processing and language and all kind of important functions, although motor is, of course, also very, very important, but it also relates to cognition. And we started, on the one hand, with Neville wondering also about the evolution of the cerebellum, and we do see that the volume of the cerebellum has changed, in particularly in men and in men-like apes. So here we compared 34 different primate species with each other. And particularly if we focus on the ensiform area, which is cross 1 and 2, which are also regions that are implicated in social cognition and memory processing. So it's here, and it's this lobe, and we segmented this in a subset of species. And also here we see that there's accelerated change over evolutionary time in humans and big apes. And then with Katharina, we take a different, but also a developmental approach, where we also link the development of theory of mind with cerebellar functional changes. And here we see also in exactly these areas, or in highly similar areas, the differentiation between children that have theory of mind versus those that don't. And this is all, again, all relying on open data that other people have used for different purposes. And this helps us also understand, like, maybe the role of the cerebellum that acts in concert with the cortex. And like, I'm very excited about the work going on here. So yeah, summary is I do a lot of things to kind of understand this interplay between genes and environment upon the brain, and kind of understand the rules and what is going on there. And I'm looking forward to your questions. Oh yeah, I have an overall conclusion. I wasn't so happy with it on the train because it's a bit superficial. So overall, the organization of the brain aligns with broad cognitive and not cognitive functional architectures. It is shaped by the interplay of genes and environment over the lifespan, and it may inform interventions to promote mental health at the right time and in the right framework. And it's important to consider that there's always a certain dynamics between the individual and its surroundings when thinking also about the functioning that you're measuring an individual. And to me, the computational tools that I apply, and especially my team applies, they knit together this tapestry to kind of understand what is going on. That brings me to the last part. Like, we also make some toolboxes or contribute to toolboxes to like help people doing similar things as we do. And at the moment, Amin is developing QBNM, which is his way, like, according to him, everything can be turned into a GPU, and he already like made meta-analytical processes go 300 times faster. So yeah, it's very nice to see how he's also branching out in that direction. And we also wrote a statistical toolbox and Enigma toolbox for the Enigma data, and a brain space for the gradients. Yeah, and this was my team 2 years ago, I think, or 1 year or so. It has already changed, so we have to make a new photo. And obviously, I have a lot of lovely collaborators who helped me and who taught me so much. Yeah, that was kind of the presentation. So sorry if it went to many directions, but I hope for everybody there was something. And yeah, I'm looking forward to your questions.